Proteins are life's way of getting things done. From structural support to catalyzing reactions, proteins have numerous functions depending on the physiological need. In order to gain a better understanding of these proteins and their functions, we want to know that we're actually working with the protein we want without the presence of other impurities. Based on the knowledge about different proteins and their properties, and with a little help from technology, scientists and researchers have developed methods to separate target proteins from complex mixtures. This is known as protein purification. Protein purification is a fundamental concept for aspiring biochemists. Isolated proteins are always being used. From catalyzing important reactions for pharmaceuticals, to understanding the different functions of proteins through research, to cleaning up environmental messes, the applications for pure proteins are nearly endless. In our experiment for protein purification, we want to isolate alkaline phosphatase, or AP, from E. coli cells, specifically the K12 strain. AP, along with many other proteins, are found within the periplasm of E. coli cells, located between the outer membrane and plasma membrane. This is the starting point of the experiment and where we will base our first purification technique.
Since most proteins are found inside of cells, we want to use a method that would release our protein of interest into a soluble solution while reducing the number of unwanted particulates. In our case, we created an osmotic shock solution to disrupt only the outer membrane of E. coli to release the contents of the periplasm, which contains AP and other proteins. From there, what's left in the mixture are the proteins from the periplasm, including AP and the remains of the E. coli cell. To group up the cell debris, we centrifuge the mixture at 10,000 RPM to move it all the way down to the bottom of the tube. This solid mass at the bottom is called the pellet, and the remaining liquid is the supernatant, which contains AP. We want to collect this mixture for the next purification steps. After this step, we have exposed AP to the mixture and removed the E. coli cells, but this is only a fraction of the process.
For our second purification step, we take advantage of a protein's tendency to denature and fold when heated up to high temperatures by breaking the intermolecular forces that hold it together. Magnesium sulfate is added to protect AP from denaturing, leaving most of the unwanted proteins to precipitate out of the solution. Similar to the previous purification step, we centrifuged all of the precipitate down to the bottom, discarded the unwanted pellet, and collected the supernatant containing our protein of interest, further purifying our mixture.
Moving on to the next step, we want to change the composition of the solution for the final purification step while retaining our protein. Normally, our protein is surrounded by a solvent shell that allows it to be soluble in the solution. Saturating the solution with a salt, like ammonium sulfate, dehydrates our protein, making it insoluble, which precipitates it out of the solution. Just like before, the solution is centrifuged to move all of the precipitate down to the bottom of the tube. Except this time, the pellet contains our protein and is less dense than the solution, causing it to float to the top after centrifugation was completed. We carefully pipetted out the unwanted supernatant and dissolved the pellet in dialysis buffer to prepare for the dialysis. The dissolved protein mixture was then transferred into a dialysis tube, which was subsequently suspended in a beaker full of dialysis buffer. This was allowed to run for a couple of hours to fully equilibrate the inside of the dialysis tube with the external dialysis buffer. Let's zoom in to see how the semi-permeable dialysis membrane works. The membrane contains several specifically sized pores ranging from 3 to 30 kilodaltons, depending on the size of the molecules in the solution. In this case, the pores allow ammonium sulfate to diffuse out of the dialysis tube while being displaced by buffer ions. Our large protein is too big to fit through the pores and remains in the dialysis tube. This process can be repeated multiple times to further clean the solution. After the dialysis is complete, we transfer the contents of the dialysis bag into microfuge tubes to centrifuge to remove any particulate material and unwanted protein. After discarding the pellet, the supernatant was then filtered through a syringe to further cleanse the solution. At this point, we have successfully changed the composition of the mixture and are ready to use it and load it into the FPLC. Now, with the assistance of technological machinery, we can really isolate AP from any remaining impurities in the solution. The protein solution is run through FPLC, which stands for Fast Protein Liquid Chromatography. We use the Active Start by GE, which featured an anion exchange column, conductivity reader, and selective pump. The pump is able to move the protein through a mobile phase with varying concentrations of sodium chloride salt buffer to run through the anion exchange column. The anion exchange column is a form of liquid chromatography where the column is filled with beads that hold positively charged ions to bond with negatively charged molecules moving through the column. Negative ions in the mobile phase, including our protein AP, will bind to these beads. As the conductivity increases, chloride ions wash off our protein into our fractions. When we plot the conductivity and absorbance, we can see that the conductivity increased over the run, and there is a huge spike of absorbance under one of the fractions. The fraction at this conductivity contains our AP. And after all of our hard work, we have successfully purified alkaline phosphatase from E. coli.
Overall, we should see a decrease in total protein and a lesser or equal amount of protein activity after each purification step. The small decrease in activity is due to the inevitable loss of our protein when performing a purification of any sort.
Generally, a protein assay, like the Bradford assay used in our experiment, would be performed to quantify total protein, while the kinetic assay would be used to measure the enzyme activity. Together, we can use this data to assess our protein purity. In addition to this, we can also measure our protein purity using SDS-Page. We can visualize our gel using methods like silver staining, Western blotting, and immunoblotting. Based off of their standards, these methods give us the molecular weights of the proteins in each purification step. As the mixture becomes more pure, we should see higher quality bands of our protein.
Thanks for watching. I hope you all enjoyed our video and gained a better understanding on some of the methods of protein purification. Be sure to comment the video, like the video, and subscribe for more videos like this one. See ya!